Great. Well, let's move on to the tutorial. So I have basically 4 different things that I want to go through, kind of short things. So the first thing is this interspecies example. So by interspecies, I mean that you have 2 strain or 2 species, and you want to compare a certain gene between the species, and you want to see if it's how it's evolving. So if you look at the file, the .fa file contains the protein sequence, the 2 protein sequences that have been identified as homologs between 2 strain variants or species variants. I forget what this is from. And then you also have the nucleotide sequences. So as you can see with the nucleotide and the amino acid sequences, you have the same header types, and this is the same name headers, and this is important for PAML to figure this out. So the first thing we need to do is we actually need to align the protein sequences, and we're going to do that by running muscle. It's a really easy program to run. You basically need 2 arguments, and that's in and dash out, and we're going to name this just .fa, and there it goes, and it's done. So we have the file right there. So that's the protein alignment. And then this is the nucleotide sequence. So we can go ahead and we can provide these files to PAL2NAL. So PAL2NAL here is installed in this virtual environment. And if you print the print screen, we'll see that there's 2 positional arguments. There's the peptide alignment, the nucleotide FASTA files. And there's a bunch of options. Like for example, you can have the output in clustable PAML FASTA codon format, and I like to do FASTA. And so the command looks like this. I'm gonna copy and paste it from my walkthrough. Okay, so dash output FASTA, and you want to make sure that you redirect this to a new file, because by default this prints out the FASTA sequence to your terminal. So we're going to redirect it, and we're going to call this file .codonalign.fa. Another important thing is there's a dash codon table option here, and by default it uses the universal code. But if you're looking at sequences from vertebrate mitochondrial DNA, for example, or yeast mitochondrial or ciliate, you want to be sure to provide these alternatives. Right, but this, for the purposes of this presentation, I'm just going to go with the universal code. We're going to run that, and there it goes. It's really fast. And that's it. So there we have our 2 files. Actually, we have the 1 file that we need. And I have this CodeML file here ready to go in this directory that I've prepared before I started. So the seq file is going to be the name of the codon alignment that we just made. And then the output file, we're going to name MLC tree, and this is the name of the header. So to run that, it's really simple. So the hard part is actually making this CodeML file and inputting all the parameters and all the sequence files. And to run it, you just type CodeML, and then you provide the 1 positional argument, CodeML-cl.clt, and then there it goes, it's done. So it's really easy. The difficult part is it's really hard to iterate through this, because if you just have CodeML, it's not going to do anything. So you need to provide it with the control file. And we'll see that this command resulted in generation of a bunch of files, actually. It's kind of not that convenient, but we have our main output, MLC tree, and we're going to take a look at that. So there's a bunch of stuff in this file. There's the actual codon alignment with the gaps. As you can see, I kind of just randomly chose 2 sequences from 2 genomes that I've been comparing recently, and there seems to be that something weird occurred here, some kind of deletion or something happened. I'm not sure what this is, but there's a bunch of gaps. There's another alignment with no gaps. It prints out the site patterns. You get the codon usage, sort of the, you can see which codons are preferred for all the amino acids. So that's kind of cool. You could see where most of the sort of where variation in codons is as a function of position. So position 1, 2, 3 refers to the 3 positions in the codon. So that's kind of cool. This is sort of a sum of codon usage over the 2 sequences that we're comparing. So we're comparing our 2 strain variants or species variants. So if you scroll all the way down to the bottom, you'll see that it has summarized a bunch of metrics for us. We have the S, which is the total number of synonymous sites, non-synonymous. We have our DNDS. We have our rate of non-synonymous mutations, and we have our rate of synonymous mutations. So when I mentioned that PAML doesn't really perform well with a low DS, so if DS is something, if DS is 0, then I think DNDS defaults to 99 or some weird number like that, because anything divided by 0 is infinity, so it defaults to 99. So you have to be really careful when you are reading this output, or if you are figure out a way to iterate through this, as I'll talk about later. I put together a script that's available on my GitHub that sort of iterates through this. You have to be really careful to sort of keep track of where the DS is, because you want it to be within a certain range to have accuracy. Okay, so there's the interspecies example. So this is if you're comparing 2 genes of 2 different species or strains. The intraspecies example is if you're comparing paralogs of a particular in one particular genome. So, for example, I downloaded the genome of cyanobacterium, and I looked to see if there's any genes that have been duplicated within the genome. And there does appear to be one gene that appears to have duplicated multiple times, and it's present in, there's 4 paralogs of it present in the genome, right? So we have 4 genes here. So let's say we want to run CodeML DNDS analysis on those 4 genes. So let's do what we just did. We're going to run muscle, and we're going to align the protein sequences. There we have our alignment. Look at the alignment, you'll see that they're all fairly similar in size. There's some gaps, but the alignments overall look good. And we're going to run pal2nal. And there it goes. Okay, so actually it went straight to standard output, so I do need to redirect this to a file, give it a file name, and I'm going to call this codon align.fa. There it goes. So I usually just inspect these alignments to make sure that they look good. I recommend downloading these alignments if you're working off a server and visually inspecting using a program like AliView. There's other programs out there like TView that allow you to visually inspect alignments. I think it's really important if you want to do this kind of analysis, because if you have something weird going on, like a frame shift or anything like that, then that could really throw your DNDS values off. But here, I'm not going to do that. I'm just going to show this looks pretty good. We'll see that the gaps here are arranged in triplicates, so that's good. That means this is a bona fide codon alignment, which is what it should be. All the gaps are in multiples of 3 codons. Yeah, that's good. Okay, so then we're going to run CodeML. So the control file has these file names ready to go, and we're going to run CodeML now. It goes. Provided us with this MLC tree file. Same thing, we have our codon alignments here presented for us. We have the codon frequencies, codon bias. This is really cool if you're interested in looking at codon bias. Like some of the endosymbiont genomes that we're looking at here in Montana, in John's lab, we found that there's some really cool stuff going on with codon bias. Okay, so how does this output gonna look? Okay, so we've provided 4 sequences. So this isn't a pairwise comparison of just 2 sequences, which was a fairly simple comparison that we just went through. This is a comparison of 4 sequences. So what PAML does, if you don't provide it with a tree file and you just do pairwise comparisons, it's going to actually compare each sequence to, it's going to do all possible combinations of pairwise comparisons, right? So you have, yeah. So I think there's 6 total combinations of pairwise comparisons here, and you can see the relationship between each pair of genes. So that's pretty cool if you're interested in looking at sort of the relationship between gene clusters of more than 2 genes, for example. Yeah, so you see DS is hovering about 0.25 for all of them. There is one comparison where these 2 sequences seem to be most divergent. These 2 sequences, 276 and 101, seem to be most related to each other. Then DNDS is fairly low for all of them. Actually, it's actually pretty high for 2 of the genes. Relatively high. 0.3 is, I don't know. I don't know. What do you think, Jake? Do you think there's a, is there a
threshold?
Like, yeah, what's your thoughts on this? Because I've talked to a number of people and I've heard a number of opinions as to how to interpret DNDS.
So you're likely to have more sites with purifying selection in your gene than that are under positive selection. And so if you want to have a DNDS value above 1, that's statistically significantly above 1, you need to have recurrent bouts of positive selection, right? And so you usually only see that in something like a immune gene, for example, or something that's really evolving quite rapidly. That being said, the only way to get completely unambiguous evidence of positive selection is to have a DNDS of greater than 1, right? You can interpret different thresholds as being positive selection, but if you really want to say that something is 100% this is definitely positive selection, usually a DNDS above 1 is taken as evidence for that in the context of the specific test.
Mm-hmm.
The great thing about DNDS values is that you really only need a genome. You don't need any polymorphism data, and they're really robust to things like changes in demography and other like historical processes. Polymorphism-based tests that maybe I'll discuss next time have a little bit more sensitivity for picking up on positive selection. But you can still talk about like relative constraint, right? You might not be able to say this thing is definitely under positive selection or majority under positive selection, but you can say that, you know, it seems like relative to this other gene, there's a difference, right?
Yeah. So if you have like a strain variant and an isolate, and you want to look at the strain variant and see how it's evolving, and you see that all of the genes have a DNDS of 0.05, and then there's a subset like of 5 or 6 genes where DNDS is 0.3.
Yeah. That could be interpreted 2 ways though, right? That can be interpreted as positive selection on those genes or
neutral.
a loss of constraint, right?
Yeah.
So you kind of have to think about what makes sense biologically, I guess, in that situation.
Mm-hmm.
Because for your endosymbionts, a lot of the case, it's probably not going to be positive selection, right? It'll be loss of constraint for a lot of them.
Yeah. Yeah. Okay, that's a good point. Context.
Yeah.
Okay. Cool. Well, that's sort of the demo for making codon alignments and running CodeML. So I do want to briefly sort of introduce 2 scripts that are freely available on my GitHub that people can use if they want to iterate. Like I mentioned, CodeML is not really that easy to iterate through, given that you need to rewrite the control file every time you want to run it. So So I wrote two scripts, basically, two wrappers of CodeML. And the first wrapper takes two genomes, and it does a comparison of all genes between the two genomes that are related to each other based on a given e-value threshold. So it runs BLAST of one genome against the other, and then it generates codon alignments, and it runs CodeML, basically. So that's fairly what it does. Let's sort of see what this looks like. So because this script will generate codon alignments, you need to provide it with ORFs in nucleic acid format and amino acid format, and then reference ORFs in nucleic acid format and amino acid format. Alternatively, you could also provide it with just a query genome in contigs and the reference genome in contigs, and it's going to run an ORF finding tool, namely Prodigal, and then run BLAST and do a pairwise comparison with CodeML. So let's see what this command will look like. So I have an example here, and I took a subset of the Mycobacterium. So Mycobacterium leprae is a fascinating genome. So this is a Mycobacterium that causes leprosy. So this is an intracellular pathogen. And what's really cool is that half of its genome is actually dilapidated. It's encoding at least 50% of its genome as pseudogenes, or what looks like pseudogenes, broken genes that are under neutral selection. And as a reference data, so we're going to see if we can actually pick up on this using this wrapper script. And we're going to use as reference this genome of Mycobacterium tuberculosis. So tuberculosis is not an intracellular pathogen, and it doesn't encode nearly as many pseudogenes. So this bacterium still sort of retains more of a free-living lifestyle because it does need to survive outside of host cells, whereas leprae has been adapting for, I don't know how many years. The earliest evidence we have of leprosy is probably 10,000 years of some fossils that they think of people that they think have been suffering from leprosy, right? So it's at least that old. So in this 10,000 years of evolving intracellular within the host, the genome has been undergoing what appears to be neutral selection. So we're going to provide the query genome as the subset of the leprae genome. And I say subset because this program does take about 15 to 20 minutes per genome to run. So, and as a reference, we're going to provide the tuberculosis genome. So we do need to provide the control file. So this is going to be fed into the CodeML script. And the output file we're going to call pseudohunter out. There's a bunch of other parameters here that are really not important for this presentation, but I think these two are important. And this is what I talked about earlier when I was talking about the limitations of DNDS. We want to limit DS to a certain value to have the most accurate possible DNDS calculation. So here the default is 3. What do you think about that, Jake? Do you consider 3 as a good potential threshold, or 1? Because if you have 1, then that's basically you have effectively every single possible synonymous site has had a synonymous mutation, right? Is that correct?
I think you want to be cautious whenever you get close to saturation, right? It's not just that every time you have, like when you're fully saturated, yes, any mutation you get is going to be a second mutation that may be a back mutation. Mm-hmm. But most of these models kind of rely on this idea of like infinite sites where the possibility of getting multiple mutations at the same site is 0. And so if you're getting close to saturation, that means that you could still have potentially these multiple mutations. And so you want to be pretty careful there.
Okay. Well, then let's set it to 1 then. Let's be a little bit more conservative. And then this also needs to be adjusted to something like 0.01. And okay, threads and e-value. So for e-value, you could also be more conservative and set the e-value to be, actually let's just leave it at default, but you could play around with this on the phone. Okay, so it's going to take a minute to run. Are there any questions at this point? Okay, there it goes. So between the DS values of 0.01 and 1, it's found 67 homologs. It provides some metrics of the average DN among the orthologs, average DS, and average DNDS. So if we go out into the output directory, we'll see that it created a couple of files for us. This is the raw BLAST results from the blast between the two genomes, the pairwise blast. If we go into the DNDS analysis script, we'll see that there's a lot of files in here. For each pairwise comparison, there's a amino acid file, there's a nucleotide file, codon alignment, and the protein alignment. So once you are looking at your output, you can go back here and you can actually inspect the alignment visually. So let's click on one, for example. Let's look at the 2 gene sequences. We can see that one is substantially shorter than the other. If we look at the codon alignment, we'll see that it looks like this. Something weird happened there. This would be a potential red flag. So if you have a high DNDS for this gene, you might want to look at the alignment visually and see what's going on, whether it's a frame shift or there's some kind of stop mutation going on. Let's look at another codon alignment. There's another codon alignment. These look a little bit more better aligned with some gaps here. Okay, so yeah, so you get this, and then the summary output is like this. So basically you have your query gene, you have your reference gene, and you have a bunch of other metrics like strand. And the important ones here are, for this tutorial, they're the DN, DS, and DNDS. And then there's a bunch of other metrics that it provides, like it looks at the relative lengths of the 2 sequences. It looks at there's multiple gene fragments involved. So stuff like that also provides you with the translation and that actual nucleotide sequence. So there's some pretty detailed output there. Another wrapper script that I have is para hunter, and this looks for gene homologs within one genome. So rather than having a pairwise comparison between 2 genomes, it will look for essentially what are gene duplicates, or if there's been a horizontally acquired gene that's homologous to something that's already encoded on the genome. But this idea of divergent evolution, if 2 sequences diverge, or if they're duplicated and then one sequence might acquire a new function, you might be able to detect that with a positive selection test. So I downloaded the genome of Cyanide PC, and let's see what the help menu looks like. It's a lot more simpler. Basically, you need to provide an amino acid file. That's going to be this. You need to provide genes and nucleotide format. You need to, again, provide the CodeML control file. And this is just some metrics that are user defined. So what this program does is, rather than using BLAST, it runs mmseqs. And actually, I should include a citation for mmseqs in this tutorial, because mmseqs is a great program, and I should probably do a tutorial just on that. But mmseqs is able to cluster the sequences. It actually clusters the amino acid sequences. Actually, I'm going to print the mmseqs help page. So mmseqs cluster, and you have an option of clustering. There's a lot of different parameters here that are customizable, but the ones that I focus on is the amino acid identity threshold for clustering 2 sequences, and then the relative alignment coverage of both sequences involved in the alignment for them to be clustered together. So those 2 parameters are customizable via the -C and -M. So by default, I require a 70% amino acid identity over at least 70% of the sequence length for them to be clustered together. Okay, so let's just go ahead with the command, run that. So I made the mistake of not subsampling from the cyanobc genomes. So this actually might take a little long to run. Okay, so it's finished. Let's look at the output. There, so you have your similar analysis with the FASTA files. There's 2 paralogs or potential paralogs that are found. If we look at the codon alignment, that's what the codon alignment looks like. There's a gap there. Let's see, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. Good. Okay. Yeah, and then there's the summary file, and the summary file looks like this. It basically breaks genes down into clusters of homologous groups, right? So we can see here's one pairwise comparison. So these 2 genes, 5242 and 4984, are considered homologs with a DS of 0.0199 and a DN of 0.0202. Actually, there's no DNDS provided here, so you have to calculate yourself. This gene cluster, for example, you could see the DN and DS is 0. Actually, I think that's why I didn't include DNDS here, because in these cases, you can't calculate DNDS because these genes are identical, apparently, because there's not a single synonymous or non-synonymous mutation. So you basically have cluster of genes that are identical to each other. Anyway, and that's what the output looks like. That's my lame attempt at trying to make an iterative version of CodeML without having to rewrite the CodeML control file every time. Okay, so we're just coming up on an hour now. I'm gonna